Hello, everyone, and welcome to Know for GCSE. Welcome back if you were in the previous live stream. So, today we are just going to continue off of Biology Paper 2. We want to cover all of the sciences today. So, Biology Paper 2, Chemistry Paper 2, and Physics Paper 2. So, let's continue. So, we had left off in this topic discussing a bit about variation. So that brings us on to Mendel, and then we'll talk about evolution, which we should probably spend about 10 to 15 minutes finishing inheritance topic, and then we'll be able to move on to the next topic. So, the work of Mendel. So, Gregor Mendel was an Austrian monk, and he was trained in mathematics and natural history at the University of Vienna. So, on his garden plot at the monastery in the mid 19th century, he noted how certain characteristics in plants were passed on from generation to generation. He started finding and seeking some patterns. So, he looked at pea plants and eventually he found out that he showed diagrams. Using diagrams, he found that there were links to the height of the pea plants. Now, Mendel had shown that the height characteristics in pea plants were determined separately by inherited hereditary units. So Gregor Mendel basically used his ideas on height as well as pea plants to work out that everything has inherited hereditary units. What do we call these hereditary units now? These are called genes. Now, Mendel was really important because from Mendel's work, we reached three really important conclusions. So, number one, the first conclusion we reached was that characteristics in plants were determined by that time. We referred to it as hereditary units. But it was important, no matter the name, that characteristics were determined by these genes or hereditary units. The second thing we learnt is that hereditary units are passed on to offspring unchanged from both parents. So these, I'm going to just call them HUs, are passed on from generation to generation. So, first thing we learnt is that we have hereditary units called genes, and that's how characteristics are controlled. The second thing is that these hereditary units are passed on. And the third thing is that they can be in one of two states. So, a hereditary unit can be in a dominant state or a recessive state. So, dominant and recessive means that the dominant would show. If it was just recessive, then just the recessive would show. To be aware of those three. Now, it took a while for people to understand and actually credit Mendel's work. His work was cutting edge and new to scientists of the day. They didn't have the background knowledge of things such as genes, DNA, and chromosomes. That's why it was very revolutionary for them, and they did not believe him until after his death, where people realized how significant his contributions were. Using Mendel's work as a starting point, the observations of many different scientists have contributed to the understanding of genes today. Let's look a little bit at the history. So, in the 1800s, scientists had become very with so chromosomes are just long strands of DNA, and they were first looked at in the 1800s. Scientists were able to observe how chromosomes behaved during cell division. Then, the next big discovery was two in the 20th century. So in the 1900s. So in the 1900s, scientists realized that there were striking similarities in the way that chromosomes and Mendel's hereditary units had behaved. Based on this, it was proposed that the hereditary units that Mendel had proposed was exactly these small genes of DNA that made up the chromosomes. So, 18th century, we found chromosomes. The 20th century, scientists realized that there was a correlation between the hereditary units that Mendel had discovered, as well as the chromosomes discovered in the 18th century. And because there was similarities, they proposed that the hereditary units Mendel discovered were in fact what they called genes. And these genes were found on the chromosome. So just a quick reminder of a refresher of what we've just looked at. So, Mendel was an Austrian monk who was trained in mathematics, and after doing some experiments on pea plants, he found these units called hereditary units, in which Mendel suggested that hereditary units were firstly something that everything had that was determined by your parents and unchangeable. They controlled characteristics, and they could be dominant or recessive, so one or two states. So Mendel found from these pea plants, he found from these pea plants that there are these things called hereditary units that control the characteristics, are determined by the parents and passed on from generation to generation, and can be dominant or recessive. Then we look at the history. So in the 1800s, chromosomes were discovered, and it was so cutting edge, and it was a starting point for the observations many scientists then contributed to afterwards. So, chromosomes in the late 1800s and then in the early 20th century, they found a correlation between how chromosomes behaved and how these hereditary units from Mendel's behaved. And they supposed that these hereditary units were genes that were found on the chromosomes. Now, in 1953, the structure of the DNA was determined. This allowed scientists to go on and find out exactly how genes work. Okay, and now, in relation to history, we're now moving on to evolution. So, a lot of times they ask you, or they could ask you in the exam, to define what evolution means. So, in order to define evolution, you actually may be asked more about defining the theory of evolution. So, let's look more into the theory of evolution. So, the theory of evolution states that all of today's species have evolved from simple unicellular life forms that first started to develop over 3 billion years ago. So, let's break that down now. So, the theory of evolution states that all of today's species, right, everything living today has evolved, which means is derived from and improved and matured from simple life forms that were first developed 3 billion years ago. So that's the theory of evolution. So if you were ever asked to describe it, make sure you're doing it in three key points or four key points. So the theory of evolution is the fact that all species living today, all modern species, have evolved from very simple life forms which first arrived 3 billion years ago. Now, there is a very important there's a very important person involved in the theory of evolution, and that is Charles Darwin. So, Charles Darwin came up with really important theories about evolution. He used the observations he made on a huge round the world trip, along with experiments, discussions, and knowledge of fossils and geology, to suggest that the theory of evolution had occurred due to natural selection. So, Charles Darwin proposed that the only reason why evolution works is because of natural selection. So Darwin knew that organisms in a species show wide variation in their characteristics, which is called phenotypic variation. So phenotypic, remember, phenotype is the difference in physical characteristics of a species, and therefore they showed phenotypic variation because they looked different. He also knew that organisms had to compete for limited resources. Darwin then concluded that the organisms with the most suitable characteristics were most successful competitors and would therefore be most likely to survive. So, this idea is called survival of the fittest. So, survival of the fittest is this idea that, firstly, animals have phenotype, and because of the fact that they have phenotypic variation, that means that some will be more suitable competitors than others. And therefore, that will mean that if they're more suitable competitors, they will be more likely to survive, as well as pass on their genes. So that's the idea of survival of the fittest. So, survival of the fittest is phenotypic variation, which means variation in the physical characteristics of an organism, which means that some are more suitable competitors and are more likely to survive than others and pass on their successful organisms that survive are therefore more likely to reproduce, pass on their genes for the characteristics that made them successful. And therefore, the successful genes would pass on and spread very quickly. So the organisms, therefore, that are less well adapted would then be less likely to survive, less likely to reproduce, and over time, they are less likely to pass on their genes to the next generation. So, survival of the fittest, therefore, means that over time, only the animals and species with the beneficial characteristics will become common and the population will change. So, only the beneficial characteristics would remain. Okay, so new discoveries have helped to develop the theory. Darwin's theory was not perfect, that the theory of evolution via natural selection and survival of the fittest. It was not perfect. There were some faults in it and inconsistencies. Because what happened was the relevant scientific knowledge wasn't available. He couldn't give a good explanation why the characteristics appeared because they didn't even know and have that much knowledge about genes. We now know that the phenotype is controlled by genes. So the phenotypic variation is controlled by genes. And new phenotypic variations arise because of genetic variants, and genetic variants come from mutations. Okay, now beneficial variants are passed on to future generations in the genes that parents contribute to their offspring. The development of new species is called speciation. Okay, so let me just find the notes because there's some really good notes on physics and maths tutor for speciation. Here. Okay, so I've shared another tab, which is all about speciation. So you've got the theory of evolution. All species have evolved from simple line forms that first developed more than 3 billion years ago. Evolution occurs because of natural selection. And what happens is you have mutations occur, and mutations then cause genetic variants. But obviously, since genes weren't understood at that time, Charles Darwin just assumed that these variations were just physical, and if a mutation provided a survival advantage, which means, therefore, it would provide a selection pressure, and a selection pressure is just when it's more advantageous to have a certain characteristic than another. And the mutation will then be passed on to offspring, and over many generations, the frequency of this mutation would just increase. Now, this may cause one population of the species to become so different that they can no longer interbreed to provide fertile offspring, which means that a new species is formed called speciation. Okay, so if we were to bullet point the process of speciation, firstly, it will be important to say with speciation that over a long period of time, so over a long period of time, over a long period of time, the phenotype of an organism changes. So the physical characteristics of an organism changes. For example, how humans used to have tails and now we don't have tails. So that's an example of a physical characteristic in humans that is no longer prevalent. Now, so over time, the phenotype of an organism changes, and it changes so much. Why does it change? It changes because of natural selection. And natural selection is how over time genetic variants occur, and certain genetic variants are more beneficial, and therefore those with that survival advantage are more likely to survive and more likely to reproduce compared to others, and therefore they will become more popular. Now, sometimes speciation occurs when species change so much that eventually a new species just forms. And how can you measure a new species forming? Well, a new species forms when both of the species can breed, but the offspring they produce will not be fertile. Now, speciation happens a lot when populations of the same species change enough to become reproductively isolated, which means they can't interbreed. For example, if you've ever heard of in free science lessons, he discusses how there was two populations. So imagine there were butterflies, and initially they all lived on the same island. But imagine there was suddenly a separation, and this prevented this physical separation caused what we call, which just means isolation, that they cannot reproduce, and that then means that over time, perhaps this side of the island will develop and have certain mutations occur, And this side of the island won't have the same, and we'll have another side. So eventually, they both just adapt so much that they will no longer be able to interbreed. And that one species initially, because of the reproductive isolation, has become two species. Now, extinction is also another really key point that you need to touch on. So, extinction is when there are no individuals of a species that remains. Thank you for that comment, guys. It means a lot. And if you have any questions, also feel free to comment them. Okay. So, extinction is when no individuals of a species remains. So, no fossils, no living species live. And the fossil record contains many species that don't exist anymore. These species are said to be extinct. So, extinction, in terms of its definition, it's when there are no more species, no more individuals in a species that are remaining. Now, there are a couple of different reasons why extinction could happen. So, firstly, the environment could change, a new predator could cause them to die, a new disease could kill them all. They could perhaps be in a survival disadvantage because perhaps another group of animals have got the survival advantage, are much stronger, for example, have bigger muscles, which means that they are less likely to get the food. Um, and also catastrophic events as well that cause all of a species to die. So a couple of different reasons for extinction. You shouldn't be too um, unfamiliar with it because just think of the dinosaurs. How did the dinosaurs go extinct? We got a catastrophic event. There was also environmental changes. Now we can look specifically at the dodos. So the dodos are an example of a species that went extinct, and it's a couple of reasons why. So firstly, humans hunted them, so that could count as a reason why a new predator, humans. Another reason is other animals ate all of their eggs, which destroyed the forest they lived in, and also by them all of their eggs being eaten, they could not reproduce, so their eggs were being eaten, and also their forest was being destroyed, which is an environmental problem. So just think like that. You may get a case study similar to that and ask why they went extinct. Now let's just go back to evolution. So we looked at Darwin's theory, the theory of evolution. However, not everyone agreed with his theory of evolution. Still, people don't agree. It's very controversial. Why? Because it goes against religious beliefs about how life developed on Earth. Um, it suggests that there's not really a creator and you can't be made in the image of God if everyone has developed since then. Also, Darwin couldn't explain why these useful characteristics appeared. He had no evidence because they hadn't found the gene, any understanding of that. So there was no actual credible evidence supporting his claims, even though there is now. And also, there wasn't evidence to convince other scientists because many other studies had been done into how organisms change over time. Oh, not many other students had come. This is brilliant. Thanks. Thank you guys for your support. It means a lot. If you have any questions, feel free to ask as well. But we'll be doing chemistry and physics today if you will find this more useful or less useful. Okay, so we have Darwin. We've talked about how his ideas about evolution. We also have another key character. So Lamarck had different ideas. Again, he was another religious figure, and he argued that changes in an organism happened, and the changes in an organism during its lifetime will be passed on. For example, the parents, say the mum and the dad, the changes that happen in the mum or dad's lifetime. So imagine if the dad kept using his arm to reach out and get a cup of tea, for example, and perhaps is his arm stretched. And then Lamarck suggested that the child produced will therefore have a stretched arm because changes that occurred within the parents' life is passed on. Now, the reason why this was proved wrong was because of the fact that if you're using a characteristic more, it doesn't organism. This is an environmental change. This is not a genetic change that occurs. So because it's not anything to do with genetics, Lamarck's theory was proved wrong. Okay, now other scientists developed other hypotheses as well. So they came up with things to explain similar observations. They might use their religious beliefs to help them as well. Lamarck and Darwin had different theories about how to explain evolution. So, Lamarck's hypothesis was eventually rejected because obviously people didn't support his hypothesis. You can see it for yourself. For example, if you were to die, say if you had a pet cat, for example, and if you dye this pet cat's hair or this pet cat's fur pink, for example, or blue, then you will not see the offspring of that cat with blue fur. It doesn't make sense. And that's just common sense. So therefore, Lamarck's theory was proved wrong. Love the drawings. Yeah, I think it just makes it easier for you guys to understand. So, okay. So yeah, if you dyed your cat's fur blue, the offspring of that cat will not be blue. And that's why Lamarck's theory was proved wrong. Because if you do something in changing its environmental conditions, that does not affect the genetic conditions. Supported Darwin's idea, which is another reason why Lamarck's theories were proved wrong as well. Because remember, what did Darwin say? Darwin said that all of today's species have evolved from simple life forms. And he also said this happens because of survival of the fittest, which is phenotypic variation. And after they realized that genes existed, and genes, which was actually after when Darwin existed, and genes supported his idea because the phenotype, the physical characteristic, is actually controlled by the genotype, the genes. And if you also look at fossils, so there's other ways to prove evolution. So if you look at fossils, fossils of different ages, so also known as a fossil record, it can show how the changes occurred. For example, if you look at some really old fossils of humans, you can see that some fossils had tails and suggested that humans had tails. However, obviously now we know that we don't have tails. Therefore, you can see evolution in that way. Also, bacterial resistance. Now, you may be asking, how is bacterial resistance actually support evolution and shows that its evolution exists? Well, it shows that evolution exists because what do bacteria do? Bacteria purposely develop resistance and are able to evolve to become resistant to antibiotics in order to give them a survival advantage. This proves Charles Darwin's theory because obviously they don't want advantage. And remember, the theory of evolution states that today's species evolve from simple life forms in order to become more complex in order to increase your survival advantage. Okay, so enough of evolution. We're now moving on to the next topic, which is genetic engineering, selective breeding, and cloning. So all of the different ones. And people get confused between them, so I'm going to try and explain them as best as I can. So selective breeding. Let's start with selective breeding. Feel free to put in the comments what you think each one means as well. If you know what selective breeding is, feel free to put it in the comments. So, Selective breeding is when humans artificially select the plants or animals that they are going to breed so that the genes for particular characteristics remain within the population. So, selective breeding is only possible when we have humans. And what happens is these humans artificially select characteristics in order to benefit us. For example, say if you have cows. Okay, excuse my drawing of a cow. Okay, say if you have cows, and obviously, why do humans use cows? Humans use cows for meat. And therefore, what would be an advantageous trait for cows to have? Well, an advantageous trait might be that they are bigger, because the bigger the cows are, the more meat you have, and the more meat you can eat. So the more you can profit from it. So humans might artificially select certain genes which would cause this cow to have more meat, for example, to have a higher body mass. And this higher body mass could ensure that the cow had been selectively breeding, bred so that it helps the farmers make more money, for example. It tends to be things like getting the cow to produce more milk or getting another animal to produce more meat, like this. But it's also used for things like crops, so making crops disease resistant so that they don't die, ensuring that dogs have a good, gentle temperament. I don't know if you guys knew this, but there's actually a method in which humans can selectively breed to ensure that dogs are not vicious and not that dogs are not vicious and what you call the characteristic docile, so that they are pet. That is a method of selective breeding. So before, they used to be vicious, and then they used to selectively breed the calmer animals in order to ensure that they wouldn't be vicious and hurt people. So what are the steps in selective breeding? That's what the main thing you need to know is. So the first step is, from your existing stock, your existing animals, you need to select which organisms have the characteristics you're after. So, which organisms select? So, select the desired characteristics. For example, if you wanted a cow that produced loads of milk, select the two cows, the male cow and the female cow, that both produce the most milk, and then use them. The second stage will be breed them together. And then after you've bred them together, you're going to select the best offspring that actually contains this characteristic. And don't forget to say you have to then breed them together. You then continue this process over several generations until the characteristic or the trait is fully formed. So eventually, all the offspring will just have this characteristic. So go through that again. Select the desired characteristics or the organisms with the desired characteristics, breed them together, and then select those with that desired characteristic and then breed them. Continue this over loads of generations, and then eventually the characteristic will form and all of the offspring will have that. So what can it be used for? It can be used to improve yields, to ensure that certain characteristics are ensured. And selective breeding is actually nothing new. Humans have actually been doing this for thousands and thousands of years in order to ensure that we're most food or the most meat or the most milk. Now, the main drawback is actually the reduction. So there's also disadvantages to what we call selective breeding. The disadvantages include the gene pool. So imagine what you have is every time there are two new parents, this increases the gene pool. The reason why it increases the gene pool is because there are more genes, there's more combinations because you're your father and your mother. However, if you're breeding with the same gene, that actually causes a reduction in the gene pool. It makes it smaller. And a reduction in the gene pool then means that they're all closely related. This means that this is called inbreeding. Inbreeding can cause loads of health problems because there's a more chance that the organisms are inheriting harmful genetic defects because the gene pool is limited. Like some dog breeds are particularly susceptible to certain defects because of inbreeding. For example, pugs have breathing problems because of this. Now, also, there can be problems because if a new disease appears, they all have the same, there's not much variation, so that they can all be killed and easily susceptible. So that's selective breeding. Now moving on to genetic engineering. Now, genetic engineering is all about transferring genes between organisms in order to get desired characteristics. So I'm just going to find it on the notes here. Genetic engineering. So it's modifying the genome of an organism by introducing a gene from another organism to give its desired traits. Okay, I'm just going to find the proper processes. So, purpose of genetic engineering. Well, the purpose is to modify the genome of an organism by introducing a gene from another organism in order to give a desired characteristic. Now, because you want this desired characteristic, you're going to use special science and equipment in order to modify the genome. For example, how plant cells have been engineered for disease resistance, bacterial cells have been engineered to produce substances. So, selective breeding is a natural way in which you're just choosing the mother and father who have the desired characteristics, whereas genetic engineering is more unnatural. It's a more laboratory way of doing it, in which you're ensuring that that organism now has particular desired genes by physically injecting them in. So, in terms of the process, genes from the chromosomes are cut out using restriction enzymes. So, the best way to understand this topic is to just draw this out. So, let's share this tab. So, imagine you have the genes that you want, and you also have a virus or plasmid. So, imagine you've got your gene here in black, and then you've got your virus here. Let me label these gene and virus. So what you're going to do is the genes from the chromosome are cut out using these special things called restriction enzymes. So imagine the restriction enzymes are red. They act as almost as scissors. So imagine them being very sharp. What happens is the restriction enzymes goes to the gene, cuts out an area of the gene, like so, and then extracts this, and it does the same thing to the virus or the bacterial plasmid. So it cuts out a bit of the virus. Now, what does that do? So it cuts out the virus, it cuts out the gene. So what you have left now is a sticky piece of a gene. It's got these things called sticky ends, which just means because it's been ripped out, it's just sticky on its ends. And you've also got your virus bit. So you cut them out with the restriction enzyme. The next stage is to combine them together, combine the sticky ends together using special enzymes called the DNA ligase enzyme. So you're going to use the DNA ligase enzyme to join these two together, like so. And now a loop. So you've cut out the gene, you've cut out the virus with a restriction enzyme, you've then combined it together in a loop. And now what you're going to do is, after combining it together, you're going to place it in a vector. And what happens is, in this vector, so for example, this vector could be a bacteria cell. In this vector, this gene is going to reproduce and multiply. And you will now see that this, whatever vector it is, will then contain this modified gene and loads of copies of this modified gene. Now, as the bacteria then grows and reproduces, you will then see loads of bacteria with this relevant gene. So say if this gene was used for being resistant to bacteria, then you'll see loads of this bacteria with this modified gene. So that's genetic engineering. Going over the process again, I'll show you this slide. Physics and maths tutor has a really good way of putting it into 4 easy steps for you to remember. Now, genetic engineering is also used for things like GM crops. So you may have studied things like golden rice in your school. And golden rice is when they have genetically engineered the rice so that it has certain proteins and things called keratin, which has the same protein content as meat, so that people who are unable to access healthy meat because they could have diseases can still have protein. Therefore, there's loads of good things that genetic engineering can be used for. Now, moving on to the next thing, which is the advantages and disadvantages of genetic engineering. Let's see if it's here. Here, perfect. So the perceived benefits. It can be useful in medicine. It can also be useful in agriculture because it can improve growth rates, have modifications to allow it to grow in different climates, introduce modifications that allow it to be resistant. It can add extra vitamins, as we talked about golden rice, and also greater yields to solve hunger. Risks, however, they could have an effect on wildflowers and insects because they could start competing with the other environments. So the reason why is because these crops are infertile and these genes could then spread. And if the infertility genes spread, then that means that this becomes more widespread and more crops cannot grow. Now, growing with herbicides and pesticides can kill insects as well. And people are worried that we don't completely understand the effects, and it could lead to genetic engineering humans, designer babies, all of those things. So now we're just going to go on to cloning. Theory of evolution, we've discussed all of those. So cloning and classification, and then we're done with this topic. I'm going to just have to quickly take a break for 1 second because I need to just adjust the time of the second stream we're doing. It might be a little bit later than projected. So just had to quickly change the timing of the next live stream, because that may be a little bit later because of this. But we're almost done with this topic, and then we're on the final topic, and then we would have covered everything for GCSE Biology Paper 2. So well done for tuning in, even for 5 minutes. That would still mean that you've done some good revision. So moving on to Cloning. So, cloning is creating genetic mechanism. So, in terms of plants, there's tissue culture and cuttings, and in terms of animals, there's embryo transplants and adult cell cloning. So, let's look at the two ways plants can be cloned then. So here you can see tissue culture and plant cuttings. So tissue culture is when you have a few plant cells, and what you're going to do is you're going to put them in little petri dish, or what they call growth medium. It has all of these hormones, and what it does is just these tiny little cells grow into plants and new exact copies of the parent plant. These plants can be made really quickly in very little space, and they are not dependent on the conditions it's growing in, which is why they're really useful. Then you have the second one, cuttings. So is when gardeners take, literally snip off a little bit of a plant. Perhaps it can be the stem, and what it would do in soil, you may dip it in a bit of rooting powder, and it will then produce genetically identical copies. These plants can then be produced really quickly and cheaply, and this is a quite older, simpler way of doing it. So that's the plants. You've got tissue culture, and tissue culture can be used to preserve rare plant species. All you need is a few cells, and cuttings, it's an older method, just cut it and put it in soil and it'll grow. Now, in animals, you have two methods, embryo transplants and adult cell cloning. Now, let's go through embryo transplants. So, farm from their best bull or cow using embryo transplants. So, what you do is you take the sperm cell from the prize bull and the egg cells from the prize cow. You then use the sperm to artificially fertilize the egg. So that just means so in a lab, for example, and the embryo then develops, and then you then place the embryo into a lot of cows where they can grow into baby calves. So the good thing with this method is that if I was to grow here, so imagine what you're doing is you're literally taking egg cells of imagine the perfect desired characteristics of the female, and you're taking the sperm cells of the perfect desired characteristic of the male. Now, as you fertilize them, you're then going to get loads of different embryos. And these embryos can then go and be implanted into what we call surrogate cows. And that not only means that you're only getting one cow that's going to be, that has a possibility of having desired characteristics, like we saw in um, the previous one, in selective breeding. In this case, all of the embryos are going to definitely have the characteristics, and you're going to get loads of the characteristics. That's why embryo transplants are really useful. Now, adult cell cloning is another way in which clones are made. So what you do is you take an unfertilized egg cell, you then denucleate it, which just means remove the nucleus. So imagine this is a nucleus. Stage one is to select the nucleus and then remove the nucleus. And then what you're going to do is you're going to then get a normal adult body cell with whatever desired characteristics you want. So adult body cell. So imagine you being this person. So you are person A, and person B, which the egg cells have come from, it doesn't really matter. So person A, imagine here's your nucleus, and for some reason you want to make a clone of yourself. So you're just going to take your nucleus and put it in the denucleated egg cell. Now what will then happen is the egg is then stimulated with an electric shock. So you're going to then just zap it with an electric shock. And after an electric shock, it will then cause the egg to start dividing almost as if it's been fertilized. Fertilized with electricity. Now, this embryo, this ball of cells, can then be implanted inside the womb of the adult female. It grows into a genetic identical copy of the original adult body cell because it has the same genetic information. So, if you've ever heard of Dolly the sheep, this is exactly what happened. So, with Dolly the sheep, they took a random egg cell. It could have been hers, it could have been anyone's. And then they took one of her cells. They then denucleated this egg cell and then put her nucleus into this egg cell, caused it to divide, and therefore a clone was. Now, cloning quickly gets you a lot of ideal offspring, but you also get, as we said before, a reduced gene pool, which just means there are fewer different alleles in a population. So if a population is closely related, a new disease could just wipe them out. And also, we could see how there will be aging and age-relating disorders as well. Cloning could help preserve endangered species, however, it's possible that they wouldn't be as healthy. For example, Dolly had arthritis, which tends to occur. Some people worry that humans might be cloned in the future and believe that children could be born severely disabled. Now we're just finishing up on this topic of then fossils and antibiotic resistance and classification. So, fossils. What are fossils? I'm pretty sure you have a good understanding of what fossils are. They're just the remains of organisms from thousands and thousands of years ago. They are found in rocks, and what happens is they tend to be preserved. So they provide evidence that organisms lived many, many years ago. And fossils can tell us a lot about how much or how little an organism has changed, an organism has evolved. Fossil form in rocks in one of three ways. So there are three ways fossils can form. Firstly, through gradual replacement via minerals. Secondly, from casts and impressions. And finally, through preservation. So we said firstly through gradual replacement via minerals. So things like teeth, shells, bones, these don't decay easily. So what happens is eventually they're replaced by minerals, forming rock-like substances and that are shaped around the original hard part. So imagine you have a skeleton of a fish, and then what happens is some minerals form around this fish. And as these minerals form around the fish, it then preserves the shape and the original shape. So that's firstly the minerals. The second way is through casts and impressions. So sometimes fossils are formed when an organism is buried in a soft material. The clay then hardens and leaves a cast. So things like footprints. This can eventually leave a long history because if you step firstly in soft clay and eventually soft clay hardens, then you'll have a cast around your foot, and then you can see it in the future. Depending on how much the clay hardens, that's how long you can see it for. But you can also see things like plant roots or an animal's burrow. So it's really interesting. And finally, the last way is through preservation. So fossils can be done through preservation. So in things like tar pits where there's no oxygen, or sometimes there's these things called amber. Now, amber is a type of clear yellow stone. So amber is a type of rock called a clear yellow stone. And sometimes found in the amber are little bugs. And these have actually been preserved by this amber. And this is a natural preservation technique. And you also see this happening in glaciers where it's too cold for microorganisms to decay, and also in peat bogs where it's too acidic for things to decay. However, no one knows how life began. There are various hypotheses, and maybe the first life forms came into existence in a primordial swamp or under the sea. Maybe simple organic molecules were brought to. There were more complex organic materials, and eventually they formed simple life forms. Nobody knows for sure. And these hypotheses can either be supported or disproved because there's just a lack of good, valid evidence. Another thing is that many of the life forms were soft bodied. Soft tissue tends to decay really quickly, and fossils that did form millions of years ago, they could have been destroyed by geological activity. So, just returning a bit to speciation, making sure that we've covered all of the relevant content. So we looked at speciation here. So a species is a group of similar organisms. These similar organisms can then reproduce to give fertile offspring. Speciation is the development of a new species, and it occurs when populations of the same species become so different that eventually they just can no longer successfully interbreed. Now, isolation is where populations of a species are separated. This can happen due to a physical barrier, for example, floods, earthquakes. They could happen geographically, different climates. And because each population shows genetic variation, they eventually just have a wide range of alleles. In each population, the individuals with characteristics then make them better adapted to their environment, better chance of survival, so the allele that controls the beneficial characteristics are more likely to be passed on to the next generation. Now, Alfred Russel Wallace was a pioneer working at the same time as Charles Darwin. Hello everyone, I hope you can hear me better. I don't know, I think there was some issues with the stream, but just returning back. So Alfred Russel Wallace, as we were talking about, he was a pioneer and he worked on the idea of speciation. During his career, he independently came up with the idea of natural selection and published together with Darwin. He then prompted Darwin to publish on the origin of species, and observations made by Wallace provided lots of evidence to support the theory of evolution by natural selection. I'm so sorry about the tech issues we're facing right now. I think the internet's just going a little bit, but hopefully you can still see and connect. So yeah, we're just going to review everything we've done, sort of antibiotics. So we've talked about how for the last few decades, we've had bacterial infections that have been viral, and they've been treated using antibiotics, and the death rate has actually fallen dramatically. However, there has been some issues because of the overuse and also the inappropriate use of antibiotics, and for example, doctors prescribing them for non-serious conditions. This means that the viruses and bacteria are more exposed to the antibiotics. And the more they're exposed, the bigger the problem of antibiotic resistance becomes, because they're more susceptible to become resistant. And in farming, antibiotics can be given to animals to prevent them from becoming ill and to make them grow faster. This can lead to the development of antibiotic resistant bacteria. The increase in antibiotic resistance had encouraged drug companies to work on developing new antibiotics that are effective against resistant strains. However, the rate of development is quite slow, which means we're unlikely to be able to keep up with the demand for new drugs. And finally, classification. So classification is just organizing all the living groups, all the living animals and species into little groups. So let's just look at that now. So you can see traditionally that organisms have been classified using the Linnaean system, which is kingdom, phylum, class, order, family, genus, and species. However, as knowledge of the biochemical processes took place, microscopes improved and we realized that there could be other systems. So Carl Woolsey proposed the three-domain system using evidence gathered from new chemical analysis, such as RNA sequences, that species thought to previously be closely connected in traditional classification systems are in fact not even closely related as first thought. So, in the three domain systems, organisms are split into archaea, bacteria, and eukaryota. So, archaea are organisms that were once thought to be primitive bacteria. They're actually a different type of prokaryotic cell, first found in extreme places, so they tend to be extremophiles. Then you have bacteria. This is true bacteria. Although they look similar to archaea, they have different biochemical processes. And finally, eukaryota, which includes a broad range of organisms, including fungi, plants, animals, and protists. Now, organisms are named according to the binomial system. Now, bi means two, so it's a two-part Latin name. The first part refers to the genus, and the second part is a species. For example, humans are known as Homo sapiens, Homo being our genus, and species being sapiens being our species. The binomial system is used worldwide, and it prevents the barriers of, country and language barriers as well. Evolutionary trees are really important to show the relationships between different species and common ancestors, and that is it for the second biology topic. Unfortunately, we did not get to go over the final topic for biology, but I would definitely say that we went over the two harder ones, and I'm glad we did because I know that a lot of you find these live streams helpful. I'm going to go to do chemistry and physics today. They'll take a lot quicker because of the content. But thank you for watching and feel free to join the chemistry happening soon.